Welcome and thank you for joining us for our computational chemistry webinars about powerful collaborative research tools. Today's webinar, Challenging Drug Discovery Project, Cloud Computing to the Rescue, is the second of our NuChem Science case study series. Over the next hour, we'll explore the tools and a real-life case study where the application of Mo's software and GridMarkets cloud platform was instrumental in bringing new energy to a project suffering due to an overly complex chemical matter. My name is Wendy Neiman, and I'll be your moderator for our time together today. We'll start out with a couple of brief presentations from Mark Ross with GridMarkets and Ben Hill with Oracle before we hear from our guest speakers, Dr. Mark Janes and Dr. Ivan Franzoni from NuChem Sciences. We'll wrap up with a Q&A session at the end, so please drop those questions in the Zoom Q&A tab located at the bottom of your screen. Now, without further delay, I'm pleased to introduce our first presenter, Mark Ross. Mark is based in San Francisco and is a co-founder of GridMarkets. Before founding GridMarkets, he served as Chief Information Officer for a number of financial services companies. Thank you, Mark, for joining us today.
Good morning, good afternoon, good evening, everyone. Thanks for your time. I'm going to give you a quick overview of GridMarkets. What we do, basically, is we run Mo simulations at scale. We also support Amber, NAMD, and GROMACS, and a few others to come. But I'm going to be talking mostly today about the Mo service that we offer. The features of that service are, firstly, you can submit from the Mo user interface directly, or you can use a command line interface. You can scale using as many Mo tokens and machines as you want, regardless of how many machines you have locally or how many local tokens you have. You can also run your simulations in parallel. You can run multiple simulations at the same time. It's a pay-as-you-go model, so you pay only for your usage, or we also have a pay-by-the-period model, which Doyle will be talking a little bit about, and it's all-inclusive, so no surprise charges. And it's secured. It's secured by the Oracle Cloud. Ben Hill is going to be talking about that today. We also are using the Amazon Cloud. So let's talk about how you get started. 3 easy steps. Firstly, you go to our website, pharma.gridmarkets.com, sign up, and create an account. That's step 1. Step 2, you download, excuse me, our plugin, which is called Envoy. And I'll explain what that does in a moment. And then step 3, you install Envoy into your Mo local instance and our SVL plugin. Excuse me. Once you've done that, you're ready to submit. So literally those 3 steps took you maybe 2 or 3 minutes. So let's talk about now how to submit from the Mo user interface. That plugin brings up this dialog box when you are ready to submit. So you can check things like, if you see over here, the machine count, if you want to run on multiple machines or break your job up into multiple parts. This is the dialog box that allows you to specify those things. So you enter the information here, and then you hit this submit button down in the bottom left. Easy as that. Once you've done that, Envoy, which I mentioned before, will transport your files for the submission and your job parameters, like how many machines you want, etc., to our cloud. It does that securely. Then we spin up the machines that you've requested. So if you want 20 machines or 30 machines or however many machines, we spin those machines up for you, and we allocate the Mo tokens to those machines for you. Again, no need to use your own tokens. So as many machines as you want, we will allocate that number of tokens for those machines. Once your job has been submitted and it starts running, then we'll deliver real-time updates to the Envoy dashboard, which looks like this. So it shows all the jobs that you have submitted. You can see, in this case, there's 2 jobs shown here. Each row is a job. You can then interrogate into the details of those jobs, looking at the logs, for example, of each one of those jobs. Here's an example of drilling down into a given job and looking at the Mo logs for that job. Once your submission has completed, then the results are downloaded securely, again, using Envoy to a designated directory. And in this case, this would be a local folder, but you could download it to wherever you chose. That's it. Let's talk about how GridMarkets compares to the other options, which would be, the big options are creating your own local cluster or building your own solution on AWS. So as I mentioned, the setup steps in our case are very simple. There's 3 basic steps. If you're building your own local cluster, there's many steps, very complicated. Same even with using AWS, many steps to set up your own cloud instance. In our offering, it's the only one that allows you to scale to as many tokens as you want. Again, we do that dynamically, regardless of how many local tokens you have. The local cluster option and the AWS option are limited by whatever number of local tokens you have. And likewise, the machines, the scaling of machines, obviously the local cluster is limited to how many machines you have in your LAN. AWS is a function of whatever your machine quota is. And in GridMarkets case, you can scale to as many machines as you need. The setup costs for local clusters are obviously very high. There's also a cost for setting up AWS, no cost for setting up GridMarkets. You can get started for free. Likewise, for maintenance costs for the local cluster is high. For AWS, it's admittedly limited, but there's no maintenance cost for GridMarkets. And then on the usage, the cost per usage in our case is very predictable. Doyle will be talking about that in a moment. In the case of AWS, you don't know what you're going to spend until the end of the month, and it can be very hard to predict that. Local cluster actually is a little more predictable because you have control over your capital depreciation and your electricity costs and so on. So in short, the return on investment of the GridMarkets option is high because you're paying for it only as you use it, whereas the other two is less so. So let's talk briefly about the costing. I'm going to introduce Doyle. He's going to explain that to you. Over to you, Doyle.
Thanks, Mark. Well, of course, Mark nailed it. We can guarantee almost that you will see improvement in your ROI within the context of one of two pricing models, regardless of the model. Model one, you can go to our webpage, easily take a look at the table, which you're seeing on your screen now, and calculate the costs on a period basis. So that's model number one. Take a look here where you can, using 5 machines for Mo on a half-day basis is roughly $1,300. Within this period, you can use Mo for as long or as little as you would like at no additional fee. Model number two, let's go up that page and you're going to see another set of examples using the famous red ball calculator at GridMarkets. Very simple to use. Select the number of machines. Let's take it to 10. Number of hours or machine hours at 20. And down here at the bottom, you'll see the machine hour cost is $19.52 an hour at about $3,900 for the total estimated cost or the total cost. Again, whether you pay by the period or by the machine hour, you're still going to experience the benefits of GridMarkets that are embedded into that pricing model, which are as follows. One, transparency. You've already seen that we post our pricing on the page. You're not going to see any changes to those costs or additional fees other than what you've seen. The package is all inclusive. Storage, egress, compute, all of it is in a single bill that you're going to see on a regular basis. As Mark mentioned, it's predictable based upon the period or the machine hour. And this is the last feature that is very critical. It is a true pay-as-you-go model, and there are no upfront fees. Transparent, all-inclusive, predictable, nothing upfront. You pay by the drink. We're going to be able to save you and do a great job improving your ROI. Go see our page at pharma.gridmarkets.com/mo. Thanks, Mark.
Our next presenter is Ben Hill. Ben is based in Seattle and is the Global Director for Oracle Cloud Infrastructure at Oracle for Startups. With more than 15 years cloud architecture and project management experience, Ben manages a global team of cloud architects responsible for supporting clients with all things cloud related. Ben is going to briefly share with us the security Oracle brings to the table. Over to you, Ben.
Thank you so much, Wendy. Again, my name is Ben Hill. I'm a Global Director for Oracle Cloud Infrastructure. So I'm going to talk to you guys briefly about security. So when Oracle went about launching our Gen 2 cloud offering, our approach to security was deliberate and direct. We wanted to ensure that it was architected in, automated, always on, and best of all, free. So I'm going to go through some of the high points about how we went about doing this. First, more security by default. Now, what does that mean? Well, we designed for isolation, and essentially what that means is we've taken the hypervisor and put it on the network layer. This approach separates customer traffic, which provides better security and performance. And as an added bonus, it reduces the risk of hypervisor-based attacks. Also, OCI has what's called separation of duties, which means Oracle admins have no access to a customer's memory space. And as it relates to your data and associated storage services, we have full encryption in rest and in motion, as well as integrated backup for business continuity and disaster recovery best practices. Now let's chat about our auto detection and remediation capabilities, specifically Oracle Cloud Guard and Oracle Security Zones. Oracle Cloud Guard provides a comprehensive end-to-end monitoring for your cloud environments by continuously collecting and analyzing service configurations, audit logs, and other information, and reporting its findings as problems based on out-of-the-box recipes or custom security recipes created by the admin. And to complement Oracle Cloud Guard or OCG, Oracle has Oracle Cloud Infrastructure Security Zones, say that again 3 times fast, which provides a secure enclave within a customer tenancy for the most sensitive workloads where security is mandatory and always on. This helps ensures compute, networking, storage, and database resources comply with your security principles, such as always on encryption, no public access, and automated backups. And just for reference, AWS does not offer any of these services or feature equivalents to OCI security zones. So just to give you just a little bit of an understanding of how we compare to our competitors. And again, like most of, as I mentioned before, the security services are free. So multi-factor authentication for user access, OCI Vault, which can be used to store passwords, SSH keys, and certificates. And unlike our competitors, OCI welcomes third-party tools, so you can federate identity and access management policies with your organization centralized identity providers, as well as SIM systems like Splunk and Logarithm via APIs and SDKs. All right, now I think we can go and move on to the fun stuff. I will pass it back to you, Wendy.
Thanks, Ben. Next up, it's my pleasure to introduce Dr. Mark Janes. Mark did his undergraduate studies in chemistry at the University of Montreal, in addition to his MSc and PhD thesis work. He's a serial entrepreneur who about 15 years ago declined an opportunity to conduct postdoctoral research at MIT to launch his first business in life sciences. Mark is passionate about improving human health through collaborative projects involving highly engaged individuals, in addition to building and growing businesses that are structured to create value in the process. He's currently partner, board member, and vice president of business development and innovation at NuChem Sciences, Inc. in Canada. Over to you, Mark.
Thank you, Wendy. Thank you, everybody, for participating in this event, the audience, GridMarkets, Oracle, CCG for producing the Mo software. I'm happy to be one of the 2 NuChem representatives today in this second webinar in this series. For you to understand NuChem's Sciences DNA, basically, we see ourselves as transforming ideas through science. More specifically, we're a small molecule drug discovery and chemical development collaborative contract research organization. In general, our objective is to understand your challenges through communication and deliver on our promise through our process of transforming ideas through sciences. So NuChem is a business that puts science first for results. You'll have to take our word for it. We have a proven track record, an unparalleled knowledge base, and a very strong culture of innovation. We have a collaborative engagement at every stage of the discovery process. So we'll try to give you a glimpse into that today. NuChem was founded in 2011. We have about 125 team members in Montreal, Canada, about 100 team members in Quebec City, Canada as well. And we work a lot with VC-driven biotechs, virtual companies, anything from 2 guys in an office, virtual pharma, to major pharma as well. And we offer different services such as project management, for example, help out if you have bandwidth issues to manage the multiple projects that you're running at your company. We have synthetic and medicinal chemistry services and teams. The synthetic part is catered towards offering and enabling the chemistry and solving chemistry issues for medicinal chemistry, but also for process chemistry and CMC stage projects as well. We offer in vitro biology and biochemistry, basically generating novel assays or internalizing existing assays to make sure that we are able to get reliable data. And data, of course, is what we base all the decisions of the program on. In addition to having good in vitro data, it's good to have in vivo proof of concept, efficacy, the translation. Also, we offer PK/PD models as well, and sometimes offer or use third-party services, for example, for commodity services or for very specialized services, like in this case, working with GridMarkets for a very specialized platform to support our computational chemistry efforts. We include computational chemistry at every stage of the drug discovery process, focused on small molecule drug discovery. So regardless if it's in hit finding stage, hit to lead, lead up, and we are even trying to bring computational chemistry more and more into the chemical development realm. So needless to say, computational chemistry is an integral part of the team at NuChem and the key to really bringing it all together, given the amount of data that's generating by different departments and entities and stakeholders in different projects right now. More specifically, on the computational side, which is the focus of today's webinar, we do hit identification. So basically preparing virtual databases, docking models, and running virtual screenings. So it wouldn't be computational without a structure-based design as well. So of course, generating of 3D models, binding site analysis, and docking studies are done on a regular basis. Ligand-based design is also one of the aspects that we focus on when structure-based isn't always possible. So database preparation, pharmacophore elucidation, and looking into the QSAR models. And of course, the name of the game is to establish SAR through SAR analysis. Lastly, for me today, we handle cheminformatics projects are more and more data intensive. So we do database management, data handling and sharing, and of course, clustering and filtering that data. That's so it can make sense for every stakeholder in a multidisciplinary project and team. So on that, thank you again for being part of this event today, and I'll turn it over to my colleagues.
Thank you very much, Mark. It's now my pleasure to introduce our next presenter, Ivan Franzoni, PhD, Senior Scientist at NuChem Sciences. After completing his studies as computational and synthetic organic chemist in Geneva, Switzerland, and Toronto, Canada, Ivan joined NuChem Sciences, where he applies his mixed experimental and theoretical expertise in medicinal chemistry and drug discovery programs, as well as being an active collaborator with academic groups in Canada, Europe, and China. Ivan is going to present a case study on the application of virtual screening of commercial compounds databases in the discovery of novel chemical hits that allowed to overcome the limitations correlated with a particularly challenging chemistry in a time and budget critical drug discovery program. Over to you, Ivan.
Thank you, Wendy, for your kind introduction. I'm happy to be back here in our second case study webinar. And Wendy mentioned, today I'm going to show you how the application of the Mo computational software developed by CCG and the GridMarkets cloud computing platform brought new life to a particularly challenging and time and budget critical project by the identification of novel and readily accessible chemical matter. Nowadays, computational chemistry is an essential part of any drug discovery program, especially in the first stages of the process that include the identification of novel hits for targeting the protein of interest, their chemical development into advanced lead compounds, and the final further chemical optimization leading to clinical candidates. In the previous webinar, we presented an example of the application of computational tools in the middle part of a medicinal chemistry program, the hit to lead stage. Today, we will take one step back and we will focus on the starting point of any drug development study, the identification of novel chemical scaffolds or hit finding. The project we are going to discuss today immediately presented several challenges related to the somehow exotic nature of this essentially unexplored target protein and the limited prior development of a handful of chemical modulators mainly derived from complex natural compounds. The goal of this computational study was to first improve our understanding on the binding mode of these complex molecules, and then to apply this knowledge in the identification of novel and synthetically accessible and feasible chemical scaffolds for the development of new inhibitors for the target receptor. As usual, every time we are faced with a new program here at NuChem Sciences, we first analyze the material provided by our clients and integrate it with our own assessments to fully understand the scientific background, identify challenges and potential pitfalls, as well as define goals and milestones. At the outset of the program, we work closely with our clients to gather as much information as possible and to fully integrate their vision into our planning. In this case, the therapeutic approach proposed by the client is completely novel and unprecedented. This aspect is certainly exciting for a medicinal chemist, but at the same time, we were well aware that the lack of prior knowledge may have posed several challenges in the development of new inhibitors, especially given the strict limitations in terms of time and budget allocated for this program. So after roughly 30 days equivalent of for this initial assessment, we were able to propose a path forward, including all the specific details regarding chemistry proposals, budget, timelines, preliminary IP analysis, etc. Our plan was specifically designed taking account the three major challenges we identified: the limited, almost non-existent prior studies on the protein of interest, the lack of known inhibitors for this target, and the limited, extremely complex chemical information based on a handful of natural-like compounds, mainly obtained via biosynthetic approaches, and very simple analogs that were obtained via transformation, such as acetylation of hydroxy groups, reduction of carbon moieties to corresponding alcohols, or hydrolysis of simple ester functionalities. Over a few months, our medicinal chemistry team was able to successfully synthesize few explorative but structurally complex molecules inspired by the limited information reported in the literature. During this initial chemistry development, several practical synthetic challenges were evident. Even after several rounds of chemical planning and brainstorming sessions, each compound required a complex multi-step synthesis, usually between 8 to 15 steps per each compound. In addition to this, a late-stage functionalization approach was not possible, and this is usually the best and fastest way to prepare a library of compounds. And in addition to this, the particularly low synthetic modularity for these compounds severely limited the possibility to carry out a comprehensive SAR campaign. Despite all these challenges, our chemists prepared few compounds that were subsequently tested in the enzymatic assay development in-house by our biology team, showing inhibitions activities in low sub-micromolar concentration ranges. Although the initial biological results were quite impressive and extremely encouraging, the chemistry side of the program was in urgent need for new directions in order to improve the synthetic turnover, prepare more compounds for testing, and to realign our approach to a critical time-dependent budget. In order to do so, and to revitalize the project, we decided to apply our computational expertise in the identification of novel, more chemical and synthetically accessible scaffolds via a focused virtual screening campaign. Our plan for this rescue computational study followed a quite standard approach. At first, we needed to prepare a model of our target protein for the docking study and identify key requirements for the binding of small molecules to the receptor. At the same time, and based on this analysis, we then proceeded in the selection of commercial compounds to be evaluated in the subsequent virtual screening. And finally, the docking results were analyzed and to identify suitable candidates for testing in our biology laboratories. As it was the case in the previous webinar, also for this target, no public crystal structures were known for the human isoform of the protein. We needed to generate an homology model. Using the Mo utilities and online resources, we first needed to identify suitable templates for the generation of a 3D model structure. The analysis of the sequence identity led to the identification of two potential non-human candidates at 38 and 22% sequence identity for the full-length protein, going up to 84 and 96% for the binding pocket residues. For both templates, a few good quality crystal structures in the hollow state were available, allowing us to apply the homology model builder in Mo to generate our docking model. The different models built by the software then analyzed in terms of geometry, and the final model were further refined and prepared for docking. So, with our homology model in hand, we next turn our attention to analyze the binding site and setting up a docking protocol for the evaluation of commercial compounds. The binding site of the target protein is characterized by the moderate exposure to the solvent and a good definition as shown by the low B score and good electron density in the original co-crystal structure template. The analysis of the pocket via the electrostatic mapping in Mo highlighted the presence of a highly lipophilic site. This observation was further supported by the few literature precedent and by the results generated by our biology team. This seemingly critical feature was included in our docking study as a pharmacophore query to ensure the proper filling of this sub-pocket by an hydrophobic moiety from the ligand. In an attempt to validate our model, we tested several docking protocols and on the basis of literature and internal data. The best results were obtained by the combination of the pharmacophore query, induced fit calculation, and final scoring of the predicted poses using the GBVI WSA delta G model. While induced fit docking allows for a small relaxation of the pocket residuum during the minimization step, thus improving the fitting of the pose in the binding site, it also significantly increases the overall time required to complete the docking calculations. We next turn our attention to compile a database of commercially available compounds for the virtual screening. Once again, we retrieved the in-stock MolPort database that includes more than 8 million compounds available at many different suppliers from all around the globe. The full catalog was first reduced to 5.4 million compounds simply based on a drug-like molecular weight below 400 and subsequently analyzed to identify the most relevant compounds for docking. For this purpose, based on our prior analysis of the binding site and the data generated at NuChem, we generated specific small SMILES query that resulted in the final selection of a bit more of 12,000 commercial compounds to be evaluated in our docking study. So now, with our database of 12,000 commercial compounds, homology model for our target protein and a docking protocol in place, we were in the position to run the docking calculations. The easy-to-use plugin integrated in the Mo interface allowed for a simple and secure transfer of the batch calculations files from our workstation to the GridMarkets cloud platform. Once uploaded, the calculation started immediately, leaving our Mo site license available for other studies. The docking of the 12,000 compounds was completed overnight, and this is especially remarkable considering that we carried out a significantly more time-demanding induced fit docking calculations. Once completed, the docking results were downloaded via the secure Envoy portal to our local machine, and we began our analysis. So the detailed analysis of the predicted binding poses on the basis of their docking score and visual inspection led to the identification of 240 compounds of interest. That represents a 2% of the original data set that was submitted for docking. In total, the overall time required to submit, run the calculations, and analyze the docking data took less than a day, most of which was essentially an overnight calculation. So the 240 compounds identified by virtual screening were proposed to the client, and after additional evaluation, 41 were purchased and tested by our biology team. We were glad to see that out of the 41 compounds, 12 compounds showing a single point inhibition of more than 30%, and 3 compounds even above 60%. The last 3 examples were then submitted to a full dose response curve determination, showing inhibition activities up to 5 micromolar, a good result for a neat compound. So although the molecules previously synthesized at NuChem showed IC50 values well below 500 nanomolar, the results shown here are extremely encouraging due to the fact that the chemical scaffolds we identified in the present docking study are synthetically readily accessible and are characterized by a high level of modularity, exactly what we need for a well-paced SAR campaign. Furthermore, these chemical scaffolds are unprecedented inhibitors for this target protein, bringing an additional level of novelty to the program. Overall, the results obtained by our virtual screening provided the boost required for the synthetic chemistry team, and our current efforts are oriented to the preparation of several analogs of these hit compounds. So in conclusion, the application of the comprehensive MO software and the computing power provided by the GridMarkets platform was instrumental in the identification of novel, unprecedented, and synthetically feasible compounds acting as inhibitors for the target protein. The study I presented today took an equivalent of 2.5 months work among computational and biological studies carried out at NuChem. This time includes the preparation of the docking model, the time required to run the calculation of the GridMarket clusters, the analysis of the data, as well as the idle time required for the compounds to be received by our biology team. In view of the stringent limitations in terms of time and budget, the quick identification and efficient development of readily accessible chemical surely represent a game changer in this drug discovery program, allowing for a more productive experimental SAR campaign toward the design of lead candidates. So with this, I finish my presentation. So Wendy, it's back to you. Thank you very much.
Thank you very much, Ivan. Now we have time to take a few questions from the audience, and looking through the questions that we have here, it looks like the first question is for you, Ivan. And the question is, in the docking study, you mentioned that out of the final selection of 240 molecules, only 41 were tested. Is there a reason why not all the 240 compounds obtained from the docking study were purchased for biological assays?
So that was indeed the discussion we had with our client. So I mentioned a few times that the budgeting for this program was extremely critical, and I could not stress enough the word critical. So the 41 compounds were purchased based on further refinement of the docking studies and internal discussions. But given the good results we obtained with these compounds, the idea now is to explore more chemistry around these scaffolds, and also to go back to revisit not only the remaining compounds from the 240 molecule selection, but also maybe to explore close related analogs via different type of approaches. Usually we call them catalog SAR, so to identify other compounds of interest just from the analysis of chemical similarity.
Thank you, Ivan. Our next question looks like it's for Mark James. Mark, the question is, should all small molecule projects include computational chemistry at every stage?
I'm a bit biased. I would say yes, but that's not my full answer. I think it really helps bring a good dose of reality to different programs. I think you're going to create chemical matter in a more structured and realistic way if you use computational, and you're going to know, hopefully, if the model is validated and you run all your checks and SAR correlates, you're going to know which molecules that don't have enough promise, to be made synthetically. So I think it's even greener to do chemistry to some extent and include in silico most of the time. So yeah, long story short, yes, it's valuable at every stage. But it has to be validated and meaningful. For some projects, it doesn't apply, so we run the checks, and if it doesn't apply, we don't use it.
Excellent. Thank you, Mark. Looking through, we have another question for Ivan. Ivan, how did you validate the docking protocol to molecule selection?
That's always the question when you start the docking campaign. What is the level of confidence that you have on your model? So the best way to do that usually is to benchmark your model, your docking approach with the experimental data known from the literature, or sometimes it could be the client or data that we generate in-house. Unfortunately, in this case, there was some level of uncertainty on the quality of the model due to the fact that really this protein it's extremely understudied, and the chemical matter out there was not representative to fully obtain 100% confidence on the model and the docking protocol. So in this case, we were not able to fully optimize and validate our docking protocol because we had only a bit more than, I think that were 15 to maximum 20 compounds with the activity data that we could use to validate the model. So of course, that's in any docking campaign, there is never the model. You start with a model. We did our best with the model we had. We managed to obtain very interesting hits. The next step would be to integrate new results obtained by our biology team to further refine the model in an iterative process to make it better and better and more predictive.
Thank you, Ivan. It looks like we have one more question, and we might be able to take one more. So if you have an additional question, please drop it in that Q&A at the bottom of your Zoom screen. But it looks like this might be our last question for right now. Is in your pharmacophore query, were there other features in addition to filling of a specific site by a hydrophobic group?
Absolutely, yes. So the filling of the hydrophobic site of the pocket was clearly a requirement for the overall binding, but in order to maximize the interaction of the ligand with the pocket, there were other key interactions, mainly based on hydrogen bond matching between the ligand and the protein that were of course included in the pharmacophore model as well.
Thank you, Ivan. It looks like we have one more question. And this question is at what level can you go for the purity of your final compound following the NuChem computational model? It's kind of a multi-question question. And then the next part is, how much is it reliable for qualitative and quantitative analysis? It's from Dr. George Ramirez, researcher in drug development and analytical biochemistry.
I'm not sure if I fully understood the question. I think that is related to the so what the purity of the final of the compounds that are tested may affect the activity data and how this translates to the quality of the computational results. So of course that's one of the key questions every time, especially with the commercial libraries. So we all know that in several cases, some of these compounds may not be at the top of the quality we want, but for an initial screening, usually it's more than acceptable. And of course, one of the first tasks of our chemistry team, after we obtained the preliminary IC50 values, was to synthesize again the exact same molecules in our labs, so we can have all the analytical data to be sure that the compounds are extremely pure, and then retest them again in the biological assay to determine their IC50 and compare it with the commercial sample. And just one step back, of course, in any case, every time we carry out this kind of HTS screening, all the compounds that are identified to be active, then we always test in-house the purity, usually by HPLC and NMR to ensure that the quality of the commercial compound was enough to have reliable assessment for the compounds.
Great. Thank you so much, Ivan. It looks like that is about all the time we're going to have for questions. We would like to take this opportunity to thank our presenters and you, our audience, for joining us today. We look forward to seeing you on March 16th, 2022, which was coming up quicker than we all know, for our next webinar on powerful collaborative research tools titled Protein to Hit, Easily Set Up and Run Mo Docking Calculations Without Token Limits. If your Mo counts are preventing you from running Mo simulations at scale to meet customer demands on time, you don't want to miss this webinar. Industry experts from Chemical Computing Group, NuChem Sciences and GridMarkets will demonstrate how to set up and run virtual screenings in Mo at scale, regardless of your local token or machine count. Learn how, with 4 simple steps, you can set up high-performance Mo calculation on the GridMarkets cloud and accelerate your time to results. If you would like to connect directly with today's presenters, you can email them at the email addresses on your screen. And we invite you to join our collaborative community and follow us on LinkedIn and Twitter. Again, thank you very much for joining us today, and we look forward to seeing you at our next webinar on March 22nd.